Hello, good afternoon. This is Greg Horoshovsky, and I am here for CTE 6560 to talk to you a little bit about my depth of field research. So let me go ahead and share my screen with you. Okay, and hopefully we can see that. So, my depth of field research, I teach health sciences at Southeast Raleigh Magnet High School. And my courses are not the typical state of North Carolina courses. I teach all Project Lead the Way courses. So, I just completed training for the fourth and final capstone course in the Project Lead the Way biomedical science curriculum. And so I'm going to talk to you about what I did my research on. And my research begins with a little bit of history. It begins with actually in the early 1990s with the Human Genome Project, which sequenced the entire human genome um, and was completed in 2003. You may or may not know that actually the Human Genome Project was extremely controversial at the time in the 1980s and early 90s when it was going to be done because it was felt that the Human Genome Project would distract from traditional biology, which asked very specific and very focused questions. And there was a fear that it would open the door to big biology. And as things turn out, they were actually right. That is exactly what the Human Genome Project did. Because as the Human Genome Project was being completed, it became evident that we needed much more sophisticated algorithms and computer storage solutions to store the data and then also to make sense out of it. We realized that the human genome was much bigger than we thought it was, and also much smaller. That the percentage of the genome that actually coded for genes that make proteins was really relatively very small, and most of it was what we at the time called junk DNA, but now we understand that those are the control and regulatory elements in that. And when the Human Genome Project was completed, one of the fathers of that science, Lee Hood, coined the term systems biology, which is the looking at biology the upside down of how we traditionally have looked at it. Traditionally, we've used a reductionist approach where we dissect a body and we take out the organs and we look even finer and finer microscopically, getting down to the cellular and molecular level to find the parts that make it work. In systems biology, we take the opposite approach. We take the biggest possible, widest view we can and try to understand what's happening with that. And until the Human Genome Project was complete, we had never been faced with a data set like systems biology had. So, as a result of the Human Project, or Human Genome Project, the era of big biology data or biology big data and the future of all 21st century biomedical science was born. And rapidly on the heels of the genomics project, we had the omics revolution. We realized if we could look at the full total sum of all genes and all genetic material in a person, we could also look at which genes are being expressed in which tissue at what time, which was the field of transcriptomics. And then we realized we could take snapshots in time of what proteins were present in any one tissue over time, the field of proteomics, which led to the field of metabolomics, looking at all the metabolic products of something. Then the microbiome exploded onto the scene, looking at all the microorganisms that live in our gut and keep us healthy. And these huge databases began to appear right on the heels of the Human Genome Project. And in order to make sense of that gigantic data, we had to then begin to learn how to use really sophisticated statistical analyses like hierarchical clustering and the Pearson correlation coefficient to mine the information out of the data, right? The data is just numbers. But to get usable information, we have to have a smart way to mine it. On top of that, the last omics that was born of the Affordable Care Act in the Obama administration made millions of electronic health records available for data mining so that we can now go into the electronic health records and tie that together with transcriptomics and metabolomics and so on and so forth. And as you can see, this becomes rapidly very, very big. We realized this as early as 2009. Pavel Pevsner in Science magazine has quoted as saying, computing has changed but biology hasn't kept up, and specifically biology education hasn't kept up because in order to mine these data sets, which will dominate this century of biological research, most of us, myself included, didn't get the kind of formal bioinformatic training we needed. Now, I have subsequently acquired that in my PhD program, but most collegiate biology doesn't even begin to address bioinformatics, how we mine these data. And this includes our biology teachers, both at the collegiate level and in the secondary education, and even in middle schools. We really need to start introducing these concepts of big data in biology. Recently, the issue with regard to teachers has somewhat had a solution put out there, which involved creating what we call an sorry, that's the alarm at my school. An incubator, which is called Nibbles. It's the Network for Integrating Bioinformatics into Life Science Education. And it is an online, free of charge clearinghouse for educational resources in bioinformatics so that a science department at a high school or a college could make some modules using these to begin to prepare their professors to understand bioinformatics, and then also hopefully begin peppering that and seeding that into the curriculum. The reason this is so important is if we look at the United States Bureau of Labor Statistics in STEM jobs. Now, this is from 2010 to 2020, but it is very similar for 2020 through 2030. I took this from one of the slide presentations in our course, but it's a nice graphic representation that even for the next 10 years, from 2020 on to 2030, biomedical engineers are probably going to be one of the most rapidly growing fields in the STEM sciences. And this is because of this phenomenon of the big biology and all of the omics. And now taking the promise of modern medicine and tying cancer data with metabolic data, with doctors' records and outcomes from clinical trials, we can now begin to put this together. But we've got to be able to mine it, and we've got to train our scientists to use computers. But this also now has implications back into high school where I teach, right? Because if we are building biology and it's growing by leaps and bounds, every couple of days we double the amount of information that way. If it's growing so rapidly that in order to educate the next generation of scientists, we have had to push the teaching of molecular biology, the tools of biotechnology, back into the high school. I teach curricula, Project Lead the Way, and in the courses I teach, these are just a smattering of some of the subjects I teach. I teach DNA sequencing, bioinformatics, how to mine the databases at the National Library of Medicine, DNA microarray analysis for looking at cancer data, polymerase chain reaction to amplify DNA, CRISPR gene editing technology and gene therapy. We learn about nanotechnology. My students this year will be cloning in my classroom. They will clone a gene as part of one of their assignments. Here's the thing. Because there's this push back where we have to educate people earlier, high school students are being asked to learn about techniques like CRISPR and nanotechnology that they just don't have the biochemistry prerequisite to understand it. And this is because most high school students have had a freshman biology class. Maybe if I'm lucky, by the time they get to me, they've had an AP bio, but they've had maybe chemistry and inorganic chemistry class, but no biochemistry at all, which is quite a different field. And it's absolutely essential to understand these techniques that we're teaching them so they understand some of the theory behind it. So my problem is, how do I help prepare my high school students so when they get to college, they know enough about these techniques to give them a little bit of an advantage, or at least to make them feel comfortable with what they're going to be faced, which is a tremendous amount of new material. So my brainstorming process, I used a Jamboard to do this. And the first thing I said, well, we could do nothing. We could just teach the classes as they're written and just ignore the fact that the students don't know what I'm talking about when I talk about 5' to 3' design of primers in a polymerase chain reaction. That's right over their head if I don't teach them biochem. The problem is they're going to learn in college. Well, that's the status quo. And as far as I think, that is not my job, is not to maintain the status quo. I'm supposed to push and improve things. Um, I asked the science faculty, the gentleman who has the chemistry lab right next to my classroom, Mr. Remy. I asked him, he's the head of the science department. Well, what do you think when you teach chemistry? Could you add a unit on biochemistry, or could we maybe add it to the AP biology or maybe even freshman biology? And that frankly was met with laughter. He didn't even answer. He just laughed. So that was not a way forward. Um, I thought about maybe trying to add a biochemistry course to the high school curriculum, but there just isn't room. Project Lead the Way are elective courses, and there just really is not yet the room for that course. I could then just maybe pepper little biochemistry lessons into my lectures as I go along, as they arise in the PLTW curriculum. Problem with that is very often when I'm teaching gene therapy, it hits on 5 or 6 different biochemistry issues, so that in teaching the necessary biochemistry that they need to make this happen, um, I lose sight of my lesson entirely. So my solution here on the orange post-it that I've circled is I want to create a short, fun biochemistry camp, like a boot camp for the beginning of the school year. And so here's the solution. The first 2 weeks we have of school usually don't have a whole lot. Oh, I'm sorry. Don't have a whole lot of gradable assignments going on. We work on class norms. I am sorry. My computer is going. So I would like to use those 2 days to build our class norms and go over the syllabus and go over the computer rules and build our digital laboratory notebooks. But in the process, use a lot of my old Prezi lectures, which I used to teach anatomy and physiology at a nursing school. So I've got plenty of material to teach a basic level biochemistry course, but I want to take advantage of some of these really great free resources written by other teachers out there. And one of my favorite resources is a program called Nearpod. So I've looked up and I found really fun kind of interactive lectures and quizzes and sort of formative assessments in here where we can go through. So working with Mr. Remy, our chemistry teacher, we condensed my list down to something that could be given over the course of 10 days, 2 5 weeks, or 2 weeks of 5 days of school. So we begin with just a lecture on chemical bonds, ionic, covalent, hydrogen bonding, a little bit of Lewis structures so they understand why carbon only has one line on it. Water. My students have no idea how amazing water is and how it drives most of your physiology, including the synthesis of ATP. We need to look at the difference between the 4 biology macromolecules that make life: carbohydrates, lipids, and phospholipids, proteins, and nucleic acids. We need to look at the central dogma of biology, that DNA is transcribed to RNA and translated to protein, and how proteins are put together. We need to look again using water at osmosis, which drives a lot of our physiology, and then just learn the rules. They're not typically very difficult rules, but students don't learn them. The rules of DNA replication and RNA synthesis. And then finally, one day where we're going to play around with the workhorse of all molecular biology, electrophoresis, and understand the science of electrophoresis. And I've built 2 days, days 9 and 10 are just filler days where I haven't put anything to learn, so that if one of these lessons or a couple of them go a little bit over, we've got a little bit of wiggle room in here. So how it will work is it will be in the first 2 weeks of school. So, as a matter of fact, as I'm recording this today, was the first day of school, and we started talking about biochemistry boot camp in my senior level capstone course. I'm preparing them for college, and I want to make sure that they have the basics as they go forward. So, we're establishing norms, we're building trust, and we're doing all of those things, but I'm peppering into it some kind of structure to introduce this prerequisite material that they will build on over and over again. It will be reinforced over and over again throughout the year. And again, my vision for this is that I don't want to give a lot of homework. I don't want to get really heavy into chemistry theory. I want to get them the general idea of how these molecules work together and how humans have manipulated it for to make new tools for the modern century, the medicines that we'll see. mRNA vaccines are a perfect example of this kind of work, right? So I see no summative assessments, maybe some, formative assessments in the game in the way of like a Kahoot game or a poll or something fun. Mostly just fun activities and discussions and letting them ask questions where they have misconceptions about that and just kind of giving them those materials. So when we're going through, I can say, hey, do you remember about quaternary structure of proteins? Remember how we said those subunits came together? And then we can have a discussion about restriction enzymes. So thanks for your attention. Um, I'm a day into it. It looks pretty good. I'm hoping that 2 weeks later, I will feel just as positive about this as I do today. But thank you for your attention. And here are my references. Thanks very much.